Hello, everyone. The title of my talk is Meet the First AI-Designed Organism. What does that mean? Where do they come from? What could they be their purpose? This is what we're talking about today. Let's see how we make this progress. Yes. So, first of all, what does it mean, an AI-designed organism? The origins of this new sector of science are in 2020 through the fundamental work of essentially two genius scientists, Michael Levin from Tufts University and Josh Bongard from University of Vermont, who actually designed the first AI-designed organism. That is a living organism that has been essentially optimized for certain functions through AI, through an optimization routine. And the fundamentals of that is that how is it possible to have an AI-designed organism? What does that actually mean? And what it means is actually that what the original, I would say, understanding of the biology was that genetics determines the phenotype, in fact, is not necessarily very true. That, in fact, genetics is not determining fully the phenotype, and that the space of possible shapes of bodies with the same genetics is much more vast than what you would think originally. And so that actually gives the, the opportunity for us to, if we understand how to do it, to actually control the process and optimize the process for our purposes. I don't know how this video is going to play, but those are the first attempts to, in fact, create organisms that have the same genotype, but have very different shapes. So you see, for example, on the top, a fish that has 3 eyes, with fully functioning eyes. We see, hopefully this video will play, or it's not playing for some reason. Maybe the team here can play the video, but basically in the middle, we have a worm with 2 heads, which is a fully viable animal. This is a planaria that have been created through essentially a very interesting process that we're still not fully understanding, but we're going to get there, that basically will lead us to what is called a morphological compiler, which means that if we are to specify, here you see the 2-headed worms now, excellent, that you specify the final shape of the body and the system essentially backtracks or backpropagates what should be the developmental stages of an organism for this shape to be viable as an organism, as a live organism in the once it develops. For example, we spoke at this conference a lot about intelligent systems, about organoids, brain organoids, etc. What is the interesting thing actually here is that it's not only brains that basically compute and have intelligence. There is also a concept of somatic intelligence of tissues and multicellular organisms. They have their own intelligence, not necessarily maybe as pronounced as our brain intelligence, but they do have it, and it actually is a nested structure all the way down. It's a structure of cognition and a structure of decision-making at different levels of complexity of a living system. And I hope that this video plays. No, not. All right, let's try it again. No, it doesn't play. Well, this on the right is actually a video that's supposed to show the communications that there are between cells through what is called gap junctions and calcium spiking. In fact, there is a propagation of information at the level of a multicellular tissue, even if that tissue, here you see it actually now, even if that tissue has no brain or no sort of, other purpose than being a tissue. And those type of new materials, we call them agential materials because they actually have agency, unlike, let's say, dead, devices, measuring devices, robots, etc. Agential materials have this already embedded billions of years of evolution in their sensing, in their decision-making, and essentially in the way that they interact with their environment. And for us, this is a completely new dimension that is opening itself to basically have a handle on how to utilize those agential materials in practical applications for our technology, for our planet. And what is embedded in those materials? What is embedded in the living systems? They have actually a lot of things that are natural, that you don't need to essentially reinvent the wheel and program complicated things, because they're already there. We have sensing, we have the ability to grab or sequester things, hold on to things. We have the ability to move. They have the ability to communicate between each other. They have also the ability to swarm, so create an emergent structure of cognition, of decision-making at the level of a population rather than individual organisms. And the other thing which is interesting also that Xenobots actually operate in what we call the mesoscale. It's between the microscale, like bacteria, and insects, let's say. It's a niche that has its own place in nature, and hence that's where we are interested in developing applications to actually utilize that. So, Xenobots, as we call them, why Xenobots first of all? Because it comes from Xenopus, which is a frog, and the cells of a frog can be coerced or guided towards creating new morphologies. So a normal frog, as you see on top, is a developmental, I would say, pathway of a frog genotype that results in a behavior of frogs. A xenobot is a development pathway of a frog genotype that results in a completely different behavior. And that's actually the key to the point. This is where it becomes interesting is, of course, it's a very complicated problem as to understand how a biological system works. I mean, we're nowhere near actually understanding that. However, what the hope is that we, and hope and already the practice actually, that we are able to marry the fantastic computational capabilities that we have already to our merit, to basically simulate initially the assembly of cells, optimize this assembly of cells for a certain task, and then actually create the biological system of that optimized simulation. This is what actually is happening now. This is what we're trying to exploit. This is an example of one example that has been achieved of a walking Xenobot. And again, I hope this thing is going to work as a video. On top, you see the actual simulation with a supercomputer at Vermont University of an assembly of skin cells and muscle cells. Muscle cells are the ones that contract, and the skin cells, don't know why it's not playing now, but anyway, muscle cells that contract and the skin cells that do not, and that create essentially sort of the skeleton of the thing, right? And as you see that below is, in real life, a Xenobot that has been designed by the, let's say, blueprint, which the AI found, to walk from left to right. So now the question is that this is all nice as a theoretical thing, as a curiosity, but the question is, can we design Xenobots for specific purposes to solve problems that are not solved now by current technological means. So one example, I'm sorry, I need to go back here. One example, for example, is the problem of finding very minute quantities of something in, let's say, a liquid. An example is PFAS, which is this forever chemicals that clog our planet, and there is a massive effort to basically reduce and clean the PFAS or detect PFAS in water. There are guidelines, is a $20 trillion problem per year. So here is, in that example, we are utilizing, this is a simulation, of course, simple simulation, where one Xenobot, essentially because it swims, finds such a tiny molecule in parts per billion in the water, and then essentially changes state, because it can sense it, it can change its state through specific synthetic biology constructs, and then as it bumps into other Xenobots, it communicates to them that it has changed states and the others change states. So it's almost like a chain reaction in a biological system, in a swarm. And this is the type of, I would say, transition from the micro scale to the macro scale that we are, I would say, working to exploit commercially. So I mentioned PFAS, which is a problem, recognized big problem. Another recognized big problem is mining. So there is a lot of mining going on, but the actual yield on mines is, could be much more enhanced by in fact, finding those metals in the water, in other things that are, or in the acid that comes after the mine. And another thing which we're very interested in developing is a new class of biosensors through those Xenobots, which actually measure some features of an ecosystem. Like, for example, in hydroponics. In hydroponics, you have to have a specific balance of chemicals for plants to have maximal yield. And we think that there will be some very concrete applications in that field as well. And again, the key to the story is that we're measuring things and acting on things with alive material, not dead material, and that we can use the scale amplification of swarming. So it's actually almost like an emergent transition to a different behavior to actually create those new sensors. So for this, we created a company 2 years ago called Fauna Systems in partnership with Michael Levin, with Josh Bongard, and with another partner, Randy Castleman, in the US, which we called Fauna Systems, and which we're currently building the technological stack necessary for commercial applications, for scaling commercial applications. So, for example, one of the big wins we had recently is the obtention of a grant for an automated laboratory, which we call the robot scientist, that will essentially create that feedback between the real biology and the AI or the simulated environment to optimize those beings for a specific purpose. And in fact, our business model for that is XaaS. What does it mean, XaaS? It means Xenobot as a service, because we will use essentially like a B2B model to create solutions for people, for companies that are in the field of environmental remediation and the like, as a service to create new, essentially reagents, new tools for them to solve the problems that they have at scale. On this, I would like to tell you, thank you very much for listening to me. Please watch this space. We hope to come back with more advancements in the near future. Thank you very much.